In this presentation, we will look at the impact of high temperatures, those temperatures that exceed the optimum range, and the effect on turfgrass growth and development, and thus the impact on the turf's quality. In studying plants like turfgrasses and how they respond to environmental stress, it is important to keep in mind that plants are complicated. These are just three pathways found in plants. Up in the left-hand corner, the carbon fixation in photosynthetic organisms, citric cycle, and the nitrogen metabolism cycle. I put these in not that you need to know this, but to appreciate the complexities in a plant's ability to maintain a healthy state, and how stresses can throw a wrench into plant survival and health. To begin, temperature stress occurs when plants are exposed to temperatures outside their optimum for growth and development. In addition to heat stress, two other major temperature stresses, freeze and chilling, can occur, which will be discussed in a future module. A stress subjects a turfgrass plant to a hardship. These stresses can cause an indirect or direct strain or injury, resulting in a physical or metabolic change in the plant. Plant resistance to temperature stress, or for that matter any abiotic stress, is comprised of three components. When a plant is not present at the time of the stress, this is called evasion. This would be the case for annual weeds. For example, the true annual form of Poa annua produces seed in the spring, then the plant dies prior to the summer stress period. From a management perspective, having your turf die prior to a stress is not an acceptable means of stress resistance or management. Avoidance is the primary means a turf survives a stress. For example, a turf may not be exposed to a lethal cold temperature because it's under a blanket of snow, avoiding direct exposure to that lethal temperature. Turfgrasses may avoid, for example, moisture stress by having a deeper or more extensive root system. Again, this is an avoidance mechanism. They may also avoid high temperature stress by the presence of wind, or a deep root system, or that the turf have a more northerly orientation. This list is an example of how plants may avoid a stress. Tolerance is the internal mechanisms by which a plant protects itself from a direct stress. Listed here are a few examples. We will discuss later how turfgrass plants protect themselves against a direct temperature stress. The severity of a stress, which are listed here on the left, is determined by the duration of the stress, the number of exposures, and the presence of multiple stresses. Heat stress in combination with other stresses can cause injury to warm season turfgrasses. However, the occurrence is not as common as on cool season turfgrasses. The impact of heat stress on cool season turfgrasses will be our focus for further discussion. Cool season turfgrasses have moderate tolerance to heat stress. Heat stress often occurs in conjunction with moisture stress. The most common heat stress symptoms are a reduction in shoot growth and a stoppage or loss of a functional root system. Cool season turfgrasses have moderate tolerance to heat stress. Heat stress often occurs in conjunction with moisture stress. The most common heat stress symptoms are a reduction in shoot growth and a stoppage or loss of a functional root system. It also causes some morphological changes, like a reduction in both shoot and root growth, which previously mentioned, also in the stand density and leaf size. In most cases, heat stress alone does not cause turf death. The impact of heat is often associated with detrimental changes in the plant, like this thin, spindly plants. At the time of this photograph was taken, this green was creeping bentgrass. Under South Florida weather conditions in July, which is hot and wet, the bentgrass was thinning and lacking any recovery from wear. You can see the discoloration of the turf from the extended heat stress period. In this case, this mix of poa annua and creeping bentgrass has been subjected to a prolonged high temperature stress. The extended temperatures above normal have made this putting green and collar susceptible to wear and traffic injury. This picture was taken during the summer at a golf course in Sydney, Australia. Again, the extended high temperatures have predisposed this creeping bentgrass green to wear injury, and most likely moisture stress has occurred, which in conjunction with high temperatures have increased the severity of injury, primary to the cleanup pattern and color of this green. You may wonder what this gentleman is doing. What this guy is doing is spreading grass clippings on the dead areas of turf to make it appear green. You cannot see it, but there is a helicopter in the far top right-hand corner, which is filming this golf hole for television broadcast when this course hosts a golf tournament. The helicopter had to fly multiple times over this golf hole to film it because the clippings kept blowing away due to the swirling helicopter blades. So they eventually wetted the clippings to keep them in place. This picture was taken approximately 20 years ago. Now, if this were to be done, you would probably spray the green with a green pigment or dye to color the injured areas of the green. Two types of heat stress can occur. Indirect or chronic high temperature stress is the most prevalent. Temperatures above the optimum can result in a decline in turf growth and loss of energy in the plant through a reduction of carbohydrate levels, and indirect heat stress can cause root loss or death. Direct or acute temperature stress occurs at superoptimal temperatures that are lethal to the plant through the disruption of cellular functions. Soil temperatures above the optimum are more detrimental than air temperature. When average daily soil temperatures exceed 70 degrees Fahrenheit, 50% or more of the root system of cool season turf grasses can be lost. In a controlled study at Rutgers University, the researchers exposed creeping bentgrass plants to increasing soil temperatures while holding the air temperature constant at 68 degrees Fahrenheit. Above 21 degrees centigrade or 70 degrees Fahrenheit, they observed a decline in root mass, length, and activity, which continued to decline with increasing soil temperatures. In a field study at Ohio State University, where root growth was measured using an underground root observatory called a rhizotron, a decline in root elongation occurred when soil temperatures exceeded 20 degrees centigrade or 70 degrees Fahrenheit. In a more recent study, we looked at multiple creeping bentgrass cultivars maintained under putting green conditions and found when the average soil temperatures again exceeded 20 degrees centigrade, root mass declined in all cultivars. Root growth did not recover until temperatures moderated in the fall. Indirect temperature stress, as previously mentioned, is the primary heat stress turf grasses face. However, with increased pressure to deliver quality turf surfaces, the potential for turfgrass plants to be exposed to direct or acute stress increases. Direct temperature stress causes injury to cell membranes and proteins. Turfgrass plants do have internal means of surviving a direct high temperature stress. One way to look at direct temperature stress is to ask the question, how do plants survive a lethal temperature? In a series of studies, we looked at the response of turfgrass plants to lethal temperatures. In this study, perennial ryegrass seedlings were exposed to a series of treatments. The two seedlings on the left were grown at a constant 72 degrees Fahrenheit for 7 days. Notice the relative good shoot and root growth. The two seedlings on the far right were grown at 72 degrees Fahrenheit, but on the third day were exposed to a lethal temperature of 131 degrees Fahrenheit, then returned to 72 degrees Fahrenheit for the duration of the study. These seedlings were killed by that 1-hour treatment. The middle two seedlings were grown at 72 degrees Fahrenheit, but before receiving a 2-hour exposure to 131 degrees Fahrenheit, on the third day, they were given a 1 hour acclimation period at 99 degrees. After the heat stress, the plants returned to 72 degrees Fahrenheit for the duration of the study. The seedlings experienced a reduction in shoot and root growth compared to the control, but were alive even with twice the exposure time at 131 degrees Fahrenheit as the two seedlings on the right. Just as a note, 22C in this picture represents 72 degrees Fahrenheit, 37 degrees centigrade, which is in the middle two seedlings, is 99 degrees, and 55 is a lethal temperature of 131 degrees Fahrenheit. So these numbers are in centigrade. In a similar study, we looked at protein levels within perennial ryegrass plants subjected to similar temperature treatments as described previously. In this case, the two lanes on the left were perennial ryegrass plants grown at 82 degrees Fahrenheit, which is 28 degrees centigrade. Notice how dark the lanes are. The darkness is a result of various types and amounts of proteins that have formed. In the two far right lanes, these plants were grown at 82 degrees Fahrenheit and subjected to a 2 hour heat treatment of 131 degrees Fahrenheit or 55 degrees C. Notice how the lanes are devoid of protein. These plants, like the previous ones, are dead. Now look at the two lanes labeled 40/55, where the plants receive 40 degrees or 104 degrees Fahrenheit acclimation prior to the 55 or 131 degrees Fahrenheit treatment. The lanes have lower levels of protein, yet they are still alive. In the two lanes labeled 40 for 40 degrees centigrade or 104 degrees Fahrenheit, notice at this temperature we observed the presence of a protein that was not present at the 28 degrees C. This protein is part of a group of proteins known as heat shock proteins. Heat shock proteins are both high and low molecular weight proteins, as shown by the arrows. These proteins form during the acclimation phase, helping to stabilize other plant proteins and preventing them from unwinding once exposed to high temperatures. Once the high temperature stress subsides, these heat shock proteins degrade or disappear. The presence of heat shock proteins or classes of heat shock proteins may help determine the heat tolerance of turf grasses. At Mississippi State University, researchers determined the heat shock response in Pencross creeping bentgrass. Pencross is derived from three parents shown in the top photograph. The three bottom photos represent the three parents. The black dots represent heat shock proteins. Notice the middle photograph or parent lacks some of the proteins. This might explain why Pencross is not as heat tolerant as some of the newer creeping bentgrass cultivars. Heat shock proteins may play an important role in direct temperature stress. However, other factors are important in stress tolerances of turf grasses. More research is needed into the molecular aspects of heat tolerance in turf grasses. What can we do to minimize turf injury during periods of heat stress? That will be discussed in the following section.